Welcome, everyone, to the afternoon session. For the afternoon, we'll be shifting gears and hearing more about the role of restoration as a climate mitigation strategy and what we've learned from monitoring carbon at the site. So this is a blue carbon and biochemistry of wetland restoration session. And we have starting off is Adina Paytan, a professor from UC Santa Cruz, and her lab members. They'll share what they've learned from their ongoing project at the Slough. We are really lucky that Adina has become invested in the Slough, brought a few eddy covariance towers, and we're just learning such a tremendous amount from the work that she's doing. So we're really happy to have her here. We'll have a little discussion and Q&A after all of the talks that will be coming after Adina, so in the whole session. So thank you very much. And Adina, I'll turn it over to you.
Thank you, Monique. And there's going to be several people. Some are remote and some are here, but they know their order. And as Monique said, we'll let me share the screen. We'll be shifting gears. Okay, just 1 second. Perfect. If you can all hear me. We're going to talk about wetland restoration in terms of its role as carbon sequestration. So we talked until now about all the reasons we would like to monitor or restore wetlands, and that is for migration, food, habitat, fund, flood control, clear water, water supply, and all the other benefits that people have previously mentioned in this talk. But as Monique said, we're going to now talk about coastal wetland restoration as a nature-based climate solution for storing carbon. And again, in the introduction talks, Monique specifically mentioned that was one of the goals of Hester restoration, at least to understand or learn a little bit about carbon sequestration in wetlands. So I'm going to give kind of an overall overview of why we need to sequester carbon and why wetlands sequestering carbon, and then the students from my lab will kind of focus on their specific components of this research. So as you know, there is a lot of CO2 that has been accumulating in the atmosphere, and in the Paris Agreement, a lot of countries, actually 137 countries, signed the Paris Agreement and committed to get to net zero in order to prevent the temperature from reaching over 1.5 degrees. Now we're already past this threshold, and we're hoping to keep it below 2 degrees because of all of the negative impacts this will have to the ecosystem and to people. The US is not part of this agreement anymore, but the rest of the world is moving on. And actually, California is still trying to get to net zero by 2045. And when we talk about net zero, what do we mean? In order to get there, we want to have the amount of CO2 that was in the atmosphere at the beginning of this century. And to do that, the way to go ahead is, first of all, we had to get to reduce our emissions. Without that, that's not going to happen. But even if tomorrow we are going to go all electric, reduce all of our emissions, there's going to be residual inputs of carbon dioxide into the atmosphere from agriculture, from ship activity, from airplanes. And in order to really reach net zero by 2050, we actually have to remove these residual emissions from the atmosphere. And we're talking about billion tons of negative emissions, or directly removing carbon dioxide from the atmosphere. So in other words, to get to net zero, we need to lower our emissions, but we also must remove emissions from the atmosphere. And there's lots of ways to do that, both companies that have technologies of direct air capture and all sorts of other things. I think the one most people are familiar with when you take a plane, they ask you if you want to contribute this much money, and that usually goes to planting trees and forestation. But in addition to all of these specific ways to get CO2 out of the atmosphere, we can actually work with nature, and this is called nature-based solutions. And the United Nations Environmental Program calculated that if we want to get to net zero stay below 2 degrees Celsius, we actually have to use nature as part of the solution. And working with nature means that we want to protect, manage, and restore natural ecosystems that capture CO2 naturally from the atmosphere. There's other reasons to do that, lots of other co-benefits by doing that, but climate mitigation is one of them. And working with nature, while it cannot but on itself solve or remove all the needed CO2, there's two advantages of using nature-based solution. A, they're shovel-ready, meaning we can do them right now. It is not dependent on technology that hasn't been tested, and it actually can remove, if done globally, about 30% of the amount of carbon we need to remove. So it is meaningful. So what are these ecosystems that naturally store carbon? You can see here the rainforests, other forests, deserts, tundra, and the largest one on the bottom, you can see the wetlands right here, with overall being the largest, but most importantly, the subsurface or the soil carbon is larger than any of the other ecosystems. And why is that important? Because that soil carbon is sequestered or is removed for long periods of time. If it's above ground, it can burn, it can be chopped, it's more susceptible, and it's less durable. It doesn't stay there for as long as it stays in the soil. So wetlands are really important as an important ecosystem to sequester carbon. But you can see over here in the photos on the right that wetlands are very diverse temporally and spatially. They change over seasons. Different wetlands do different things. And in the table over there, you see red and green squares. This is the calculation of carbon sequestration that includes both carbon burial and emission, minus the emissions. And you can see that some wetlands, this is all data from the Delta area, from the San Francisco Bay and Delta, and you can see some wetlands over there actually are sources of carbon. That's all the red ones. Some are sinks of carbon, so they remove CO2 from the atmosphere. Some start as a source and then become a sink, and vice versa. So we really need to understand what controls this variability in order to plan how to construct wetlands in a way that they will maximize their carbon sequestration. And to do that, a committee in California came to some kind of focus areas for science investment in order to use nature-based solutions. That includes mapping, some statewide coordination and planning, using models. But in the red, this is one area that is really, really important, and that's understanding the biogeochemical processes that actually control the fluxes of methane and the fluxes of carbon from these systems, and where is the carbon going and how much of it, and what are the controls on these fluxes, because then we can understand potentially how to design a restored wetland or where to prioritize conservation and restoration such that carbon sequestration is one of the benefits that the wetland provides. So just to kind of give you the overall questions, what we need is data that will help us to predict where most of the ecosystem carbon removal happens and how does it happen so we can scale and prioritize. We need data to inform restoration that maximizes multiple benefits for people and nature, which includes climate mitigation. And we need to also understand how will climate change in the future impact this carbon sequestration or carbon removal, because we are changing the playing ground with higher temperatures or higher sea level rise, which may impact this ability to sequester. So to do that, we need to understand the mechanisms that control the carbon removal and carbon sequestration. The other thing that is not directly related to biogeochemical cycling and the carbon, but also related to carbon, is we know that wetland restoration is expensive, and we want to understand what are the good financial models to make sure there is money available, both state and private money. And one of these systems is actually listing wetlands as, like they do with forests, as systems where you can get carbon credits for sequestering carbon in the carbon markets, and that could benefit or could provide resources to money to restore wetlands, because this is expensive. But to do that, when you actually register a project in the carbon markets, you need to actually quantify how much carbon is removed. So we need to understand how to monitor this carbon. And again, in order to be able to monitor the carbon, we need to understand what are the predictors, or what processes determine how much carbon is actually sequestered. So with that in mind, my lab and the people that are working with me are doing different parts of this big puzzle, trying to understand what are the fluxes, or how much carbon is going in and out of the system, and what are the processes that determine these fluxes. And I will hand it over to actually Monique to talk next, because she's going to share some work that Beth has done before.
To remember the audio and video. Okay, thank you so much, Adina. So Beth Watson was going to be giving this talk, and she was not able to make it today, so I will present her data. I was just a field hand on this study, so I'll do my best. Beth is from Stony Brook University. She did some paleoecology work here in Elkhorn, Slough in the early 2000s, which I'll touch upon. She earned her PhD from UC Berkeley and was a postdoc at UC Davis. She was also a Fulbright Scholar and held a joint position at Drexel and the Academy of Natural Sciences before joining the faculty at Stony Brook. So I'll talk a little bit about what she did here. For the Hester restoration, she monitored multiple sites before and after the restoration. So this is in 2015. These included the restoration site, degraded control areas, which are former subsided marshes, and our aspirational high marsh reference site. And so we sampled separately in grassland, high marsh, low marsh, and mudflats, so we could calculate the blue carbon function by habitat type and then estimate the total for the Hester footprint. This is before and after restoration. So looking here, we have our reference sites, and you can see they're different in their formerly degraded sites. If you remember from before, we had a lot of lower marsh, and then in our restored marsh, we have higher marsh. So the below ground carbon sequestration, was monitored with the sediment cores, a lot of sediment cores. And this is new work that's built on Beth's earlier paleoecology work on the long-term rates of carbon sequestration that were measured from dated cores that she took from Elkhorn Slough marshes. And so we had all of this pre-carbon data, which was, I think, really instrumental in launching this blue carbon work here in the slough. So she looked at the rate of carbon deposition at the surface using feldspar marker horizons. And then above ground carbon sequestration was monitored by harvesting the above ground biomass. Gas fluxes were monitored with a Picaro gas analyzer in light and dark conditions. And so what we found, carbon accumulation at the surface was higher in mudflats. So if we look at this graph here, we're seeing deposition, and then along the bottom here, we have degraded marsh, restored marsh, and reference marshes. And in the degraded area, we have grassland ecotone, our high marsh, our low marsh, our mudflat, and you can see how much carbon was sequestered in those mudflats. So that was really surprising. These are also in these degraded marshes. Remember, these mudflats are former wetlands too. So those diked and drained wetlands that kind of fell apart. So these have long inundation times, lots of organic matter. And yeah, so this is pretty surprising that we saw that. And okay, so then in our restored area, this is not as much. And then in our reference area, you can see still a little bit lower than that. But I think the important thing here is that this is not a stable state. You know, a big storm event could remobilize the carbon sequestered here pretty easily. And okay, so then the below ground carbon sequestration was relatively low at Hester phase 1. That's this area in here. And so this was in 2021. So due to the grant constraints, these samples were taken pretty early on. So it was only 2 or 3 years after it was open to tide. So there was only about 25% vegetative cover, and the plants were actually relatively small. So remember, this is a really high marsh, and it will take time for all these below ground functions to mature. And of course, this is why it's always better to conserve marsh where you can. Very important. And so I guess really, in conclusion, this is why it is important to take a long-term view. So while carbon sequestration at Hester is still lower now than it was before restoration, it's much more stable in a vegetated form that can withstand the elements. And it could be one of the last marshes standing with future sea level rise. Thank you.
Back to talking about what we're doing in my lab, and we'll start with actually things that are related to what Beth has been showing. And Jess, are you there, Jess? Would you start talking? I will move the slides.
Yeah, I'm here.
Okay, I cannot, there we go, I can do it this way. Okay, go.
All right, thank you. Hi, everybody. My name is Jess Turner, and I'm going to be starting my second year of grad school as a student at CSUMB Monterey Bay. My thesis is going to be focusing on the soil organic carbon content within the marsh plains of estuaries along the coast of California. Next slide. Thank you. Soil organic carbon is accreted at high rates in coastal estuaries because the vegetated zones increase plant production, which act as a carbon source and increase overall sediment trapping. The moist anoxic soils here lead to slower rates of decomposition, allowing the plant-sourced carbon to be buried down into the sediment. These stocks can vary a lot within a single wetland or wetland type due to various climate and environmental factors such as temperature, salinity, mean precipitation, and things like that. I will be looking at those factors when comparing the soil organic carbon stocks across the different sites. Currently, its soil organic carbon data is limited outside of the CSUMB Marsh and Sacramento Delta. So this is a map of all of the sites I'm going to be sampling. The yellow points is anywhere where soil carbon has already been sampled. And the green dots are the sites I'm going to be measuring, including several locations in and around Elkhorn Slough, such as South Marsh, North Marsh, the tidal area before Porter Marsh, and around the seal bend within the main stem of the Elkhorn Slough. And I will be comparing all of the soil organic carbon data to all of the other sites I'll be collecting. So I've started collecting my samples this summer, and I've only sampled the Moroccojo Slough and the mouth of the Pajaro. A majority of my sites will have 2 to 3 stations chosen within the marsh plain, with 3 replicate cores within each. So, and that will range between the green line, which is the lower vegetation zone, and the teal line, where the tides goes for the upper inundation zone, where the soil is still wet and soft enough to core. I take these cores back to the lab. They get sliced into 1 centimeter segments, and I'll be analyzing for bulk density, soil organic matter via loss on ignition, where we burn off all of the organic matter and compare the weights before and after burning. We're able to take the soil organic measurements and estimate soil organic carbon from that, and build a curve using our direct carbon analysis using a piece of lab equipment. We will also be dating any of the cores that we can and that make sense using the 210 PB dating method. I'm hoping to have all of my sites collected by the end of the year. Thank you.
And I forgot to say, but we'll leave all the questions for the end after everybody presents, so we can have a discussion about the various projects. Thanks, Jess. Our next speaker also is going to talk about work with soil.
Hi,
Devon.
I'm Devon. I'm an undergrad in Adina's lab, and I'm looking at using biochar and organic matter soil amendments in wetland restoration, so manually changing the carbon content in those soils. First off, just a little background on what biochar is. So it's an organic matter that's been burned in low oxygen conditions to create mainly charcoal. And it's had a long history of use in agriculture, going back to Sweden, which is like slash and burn agriculture, as well as modern applications in industrialized agriculture. And it's been shown to decrease nutrient runoff and improve crop yield. And then also biochar, especially higher temp biochar, has been shown to be a stabilized form of carbon and has a lot of potential for long-term carbon sequestration. So the main question I was wanting to look at was, how does the biochar affect the carbon sequestration potential of restored wetlands? So basically, will the biochar break down with all the tidal inundation and become a carbon source, or will it be pretty inert, or will it actually act as a filter or a sponge and decrease the natural carbon loss? And then I also am looking at the effect of the biochar on pickleweed and measuring the metal content in each of the plots to see if the biochar is leaching any metals. So to measure these effects, we created 3 1 square meter plots on the edge of Hester and Yampa marshes. So one was just a control with pure soil, one had a 12% by volume biochar amendment, and one had a 12% by volume unprocessed organic matter amendment. They were placed to be at the same level of Hester Marsh, so to be a high marsh, to be inundated at the high tide, and we also transplanted pickleweed into each of the plots. And then I'm currently taking biweekly measurements of the greenhouse gas emissions, as well as taking pore water samples, which I will analyze for carbon chemistry. And pore water is the water that's held in the pores of voids within the soil. And as Mark and Aliya will be going over a little later, I can use these measurements of the vertical carbon transport, which is the greenhouse gas emissions, and the horizontal carbon transport, which is the pore water exchange with the surface water from tidal inundation, to compare the total carbon exports between each treatment. And then I'm also measuring, as I mentioned before, the nutrient and metal contents. Finally, the potential impacts of the research is hopefully it could lead to cheaper, easier, and more effective restoration. With a 10 to 30% by volume use of biochar, it greatly reduces the amount of soil you need to transport, and as soil is super expensive and heavy to move, that would make it easier. It's also, if the biochar is shown to be a successful form of carbon sequestration in the wetlands, we could get the carbon credits for the biochar that we're physically placing in, not just the effects of the wetland in carbon sequestration, and use some of those credits to offset restoration costs. And finally, if it does have a positive effect on the plant health, it could aid in the regrowth of plants in the restored marshes.
Cool. Thanks, Devon. And from the carbon that goes into the soil, we'll talk about the carbon that goes into the atmosphere with JJ.
All right. Hi, everyone. My name is JJ. I'll be a grad student starting this fall, responsible for studying the greenhouse gas fluxes from Hester Marsh. Today, I'll be talking about what these terms mean and why this is important in a restoration setting. Quickly, this picture above in our first slide is of the Hester Tower, where we mount our instruments for this. And we have this set up at 5 different sites around Elkhorn Slough. Many of you have probably seen them around. But first, let's briefly talk about greenhouse gases, why we need to measure them. So gases like CO2 and methane are really effective at trapping heat in the atmosphere, which is really what makes our Earth so hospitable to us. But excess amounts can overly warm the atmosphere, warm the land and ocean, and lead to a range of feedbacks that affect the behavior of natural ecosystems. So like Adina mentioned before, we study these natural ecosystems like wetlands to understand the drivers of greenhouse gas emissions there, and then maybe help us optimize nature-based solutions. I won't be going into the global greenhouse gas cycle, but if you want to learn more, you can scan this QR code. All right, so here is our data for the 2 greenhouse gases I mentioned in the last slide, methane and CO2. And these are at Hester Marsh since we built the towers in 2022. Briefly, I'd like to specify that in wetlands, greenhouse gases actually are being emitted and consumed at the same time all the time at various levels, and flux is the difference between the 2 opposing forces to say what is the prevailing direction and amount over a span of time. So a positive flux means more emission, and a negative flux means more consumption, or what we call sequestration. So for example, we sequester oxygen when we breathe. And each point on these graphs is a daily calculation of flux, daily emission minus daily sequestration. And the timeline over the last 3 years shows daily flux change over time. So understanding all of this about flux, let's look at these 2 graphs. In the bottom graph of CO2 at Hester, we see a nice annual pattern of flux where in yellow sections, there's clearly more CO2 sequestration through photosynthesis, which is in the growing season. And then in blue, there's more CO2 emission through respiration during the wet season, which is when the pickleweed is dormant and not photosynthesizing as much. And methane flux, on the other hand, is dominated by microbial activity. And you'll see that in wetlands, actually, methane emission is more of the trend. And so here there's not really an annual cycle, but you can see that methane emissions have gone down over the last 3 years at Hester, which is cool. Oh yes, and just so you guys know, the scale on these 2 graphs is very different. There's a lot less methane than CO2 in general at wetlands, so even though these peaks are quite high in this graph, it's pretty small. And you'll see that in this next slide, actually. In the top graph, it's the same methane flux from the last slide, except we're only looking at the last 2 years here. And we're now comparing it to another site that we measure for greenhouse gases, Pelican Marsh, which Monique and others have touched on earlier as a degraded site, mostly mudflat. And notice these 2 graphs are on the same scale. So because we don't have data from Hester pre-restoration, but we know that it was a mudflat with some similar conditions to Pelican Marsh, we used Pelican to give us a rough idea of what the data might have looked like from before. So assuming this comparison is accurate, we can make an argument that restoration steeply limits methane production, especially during spring-summer months where microbes tend to be more active. And some of my lab mates, who will present later, are working on understanding the drivers of these plumes. So they'll talk about that. And then quickly here, we're looking at a cumulative flux graph, which shows greenhouse gas uptake over time for Hester in blue, and comparing it with another reference marsh that we measure called Yampa. And a downward trend for both of these means that the sites are sequestering more greenhouse gases than they're emitting. Yampa, which has been mentioned earlier as an adjacent reference marsh, serves as a realistic target for Hester to eventually look like. And you can see that it has outperformed Hester over the last 3 years, but Hester's trend is actually catching up. And once they are about parallel to each other, we can say their net flux is about the same. I also want to specify this graph is actually representing CO2 and methane in one variable, and we call that CO2 equivalent, which is a way to combine multiple greenhouse gases into a metric by taking into account their impact on temperature. So, a lot of people say that methane has 80 times the warming potential of CO2, so it's weighed more heavily in that metric. And in summary, we know methane, which is an extremely potent greenhouse gas, and its emission are a big problem in mudflats like Pelican, potentially Hester pre-restoration. So Hester's restoration likely played a huge role in reducing its greenhouse gas impact. And also the amount of CO2 that Hester sequesters each growing season is increasing every year, and we expect that its improvement will continue as more vegetation grows in, which justifies a lot of the work that has been presented here today. Thank you.
And just to put that in context, so when we add the carbon that is buried in the soil and subtract the methane emissions, it's not clear that the mudflats are actually sequestering net sequestration of carbon better than the vegetated areas. And still going on with greenhouse gases.
Hi everyone, I'm Marco. I'm a first-year PhD student here in Adina's lab, looking kind of in a similar vein at what JJ and also Devon are looking at, kind of combining the two, but looking at greenhouse gas monitoring by land cover type. So what does that exactly mean? We're looking at least in this study, 3 different land cover types. I'm looking at Pelican Marsh, outlined in yellow, Yampa Marsh, outlined in red, and then also Hester Marsh, outlined in blue. And if you can look, I guess, within each of these, you can see that there is different land covers that take up each of the marshes. So Pelican is primarily inundated, obviously, and then Yampa is primarily vegetated, whereas Hester is primarily a high under restoration, just like covered in mud, not a lot of vegetation present. So what I'm interested in is looking at the greenhouse gas dynamics within each of these marshes, but looking not only at CO2 and methane, but also nitrous oxide. So why are these important? My main research questions are there N2O emissions coming from these different sites? And I guess, is there a specific land cover type within each of these marshes which is dominating emissions? And then even going a step further, is there a driver of these emissions? And I'm looking at doing the same types of things with CO2 and methane. Why do we care to look at N2O and methane versus CO2? Methane has over a 100-year time horizon, 30 times the warming potential, whereas N2O has about 300 times the warming potential versus CO2. So it's very important to look at all 3 of these greenhouse gases because, we are kind of interested in looking at the net ecosystem greenhouse gas balance, which we have here. So how do we go about measuring these things? We're looking to measure greenhouse gases by placing a chamber such as this one over the top of these land cover types and connecting them to greenhouse gas analyzers. So the greenhouse gas analyzers we use, I'll be using 2 of them each time I go to the field. We'll be measuring N2O, CO2, and methane, and then I'll be going out to our sites every 2 weeks to look at water, vegetated, and mudflats over each of the marshes. So yeah.
Thank you, Marco. And from the fluxes to the air, we're going to fluxes from the mud to the surface water. And Aliya, are you there?
I'm here.
Okay, go ahead.
Guys. I'm Aliya. I'm a second-year PhD student in Adina's lab, and today I'll talk a little bit about the surface water dynamics in the slough and the carbon in surface water. So Jess talked a little bit about the organic matter burial, which is the decomposition that happens after plants die and are buried. And the last couple of presentations have talked about the vertical carbon flux, which is the gas exchange from the air. But what I look at is the horizontal flux or the lateral fluxes, which is carbon that moves in and out of the slough from the ocean, mostly via tides. And at Hester Marsh, our main mechanism is tides. You can go on, Adina. And so this carbon is mostly being derived from the pore water, which Susan will talk about a little bit later. But the important takeaway is that it exists in a lot of different forms: particulate organic carbon, dissolved inorganic carbon, and total alkalinity. And if we want to understand if this carbon that is exported from the marsh is a potential sequestration mechanism, like maybe it's sequestered in the ocean, or a potential source of emissions, like maybe it is decomposed once it leaves the slough and becomes an additional source of CO2, we need to understand what types of carbon are being exported and how much of that. And this is an unanswered question in salt marshes in particular. A recent review found that this big gray chunk, 47% of the carbon that we find in our surface water, is still unaccounted for. So we don't have good data understanding how much of this is being exported and what types. So I use a couple of different methods to try and understand this. First, I'm trying to calculate pore water and surface water exchange. So understanding how much of the carbon that we see in the surface water is being derived from the pore water or being sequestered in the slough directly. And so I want to understand the percent of carbon that is coming from the wetland. Then I want to know what types of carbon exist in the surface water and in what forms. So I measure the concentrations, and then I need to know how much that carbon is moving by understanding the rate of water movement, and I multiply that by the concentrations of carbon that I find. And so I'll talk a little bit about the methods that I use for that. I use three main methods. The first is water sampling. So I have ISCO water samplers, which are automated samplers that take a sample every hour, which means that I can leave them out in the field and not worry about it and not have to stay there overnight and get a full tidal cycle. I use sensor measurements to get a sense of different water quality parameters at a finer resolution. So I have sensors that can take measurements of stuff like dissolved oxygen, temperature, and pH at 15-minute intervals, and we move these around a couple of different marshes in the slough. And then for parameters that can't be measured via sensors, I collect the water and I take it back to the lab for analysis. So I'm still early on in this research, but a couple of the take-home messages so far is that there's still a lot to understand about the controlling processes behind what drives the different types of carbon that are being exported and how much carbon is being exported, especially in terms of how land use can affect that. But some perhaps good news, and this is still very preliminary from Hester Marsh, is that the water that we're finding in Hester tends to be more alkaline than it is acidified, which is good news because if that water moves out to the ocean, it is more likely to be sequestered in the ocean, the carbon in that water, and it will also play a slight role in buffering against ocean acidification. So in this plot, you can see that the majority of the points are in the green section, which is the alkalized section.
Okay, and Aliya will continue because Zoe cannot make it today, but she will present Zoe's work because they're working together.
Mm-hmm. So Zoe is a first-year master's student in our lab, and she is doing a really similar thing, but where I look at carbon in the slough, she is looking at the other greenhouse gases that dissolve into water and can be exported to the ocean in the same mechanism. So this is important because gases, like JJ said, greenhouse gases can be produced in wetland environments, dissolve into the surface water, and then they are exported to the ocean, but they're not likely to be sequestered in the ocean. It's much more likely that they'll off-gas at some point and be an underlooked source of greenhouse gases. So if we want a full budget of the greenhouse gases, we need to understand the dissolved lateral transport. So we're looking at methane and nitrous oxide concentrations as they vary seasonally and daily. So what are the controls, like location? We're doing this in a couple of different sites: tide state, temperature, season, and time of day. Like, is it light out or is it dark out? So the methodology is very similar to what I do. The main difference is that because dissolved gases have a tendency to off-gas, leave water that it's dissolved in very easily, we have to collect them and cap them immediately, which means we do 24-hour samplings where we camp out at Hester Marsh, stay there overnight, and collect a sample every hour. And so we do that at Hester and the adjacent marsh Yampa, along with collecting a couple of other parameters that we'll use to assess the controls. And we are working on the methodology, but right now we're using gas chromatography and a Likor small flux measurement.
Cool, thanks Aliya. And from surface water, we'll move to pore waters, and Susan will come back here if she can get here. And just a second. Go ahead.
All right. Hi, everyone. My name is Susan, and I'm a graduate student in Adina's lab, and I work on the pore water part of this entire project. Oh, it does. Ah, sweet. I'll do that then. Cool. Okay. So Aliya spoke about the surface water that is in the slough, so that is influenced by tides and flooding and whatnot. And I work on a small subpart that is related to that, the pore water. So within the subsurface, you have these sediments, and there's space between all these sediment particles or soil particles. And between these soil particles, there's water, there's a little bit of air, but the water in these spaces, called pore water, is what I look at. And we care about that because the plants that take up CO2 from the atmosphere store that carbon in their organic matter, and when they, for instance, die or decompose, they get buried, and this carbon can get stored in the subsurface. Or this carbon can get decomposed into different carbon-containing dissolved molecules, such as DOC or DIC, what Aliya mentioned before, and whatever is not buried is no longer sequestered. And then from the pore water, these different carbon molecules can be transported into the surface water in the atmosphere. So all of these things together impact the net carbon balance of these systems. And one of the molecules in particular that we care about a lot is methane, because it has a very high global warming potential compared to molecules like CO2. All right. What do you think impacts the net carbon burial? Write your ideas in the chat. And while people are writing, I will move on with our method slides. Okay, so in the slough, we collect sediment cores. These are about 30 centimeters long. They're push cores. So in the top middle image, you'll see just a plastic PVC pipe that we push into the top of the sediments. And then when we've collected them, we bring them back into the lab, and then we insert tubes into the side to suck out the pore water. And in the top right, you'll see that we have attached syringes to these tubes. And after the syringe is done pulling out all of the pore water, we transfer each syringe into a separate vial so that we end up with a separate sample for each 1 centimeter depth increment of the sediments. And we take these cores in mud patches and vegetated patches. We take them at high tide and low tide and across wet and dry seasons to try and assess what might be impacting how carbon moves through the system. Because on all of these vial samples, these centimeter increments, we study what carbon species are present. And then what we get from that is something that looks like this. So this is an example of a pore water concentration profile of dissolved methane gas that we have found. I think this is from Yampa. So on the y-axis, you see depth. So this is depth into the sediments. So 0 is the top, 20 is near the bottom. And on the x-axis, you'll see the concentrations. So this is the type of data that we get from these methods. And a preliminary take-home message is that the pore water dissolved methane concentrations that I find in Pelican Marsh are much higher than those that I find in Hester. So seemingly, this restoration has been very effective in reducing methane concentrations in pore waters. I still have a lot of samples to analyze, so this is all preliminary, but so far, that seems like a good takeaway. Thank you. If you have any questions, please email me.
Cool. And as you notice, Susan is starting to look into processes that control these methane emissions and carbon burials and lateral fluxes. And some of these are mediated by microorganisms. And Leah, if you're there, go ahead.
Hi, I'm Leah. I'm currently a PhD student in Adina's lab. And my project is tracking microbial activity and community differences between restored and reference wetland environments. So microorganisms play an important role in chemical exchanges between soils and the atmosphere. My research compares reference and restored wetland sites, so Hester and Yampa, to provide insights on the impacts of wetland restoration and look at how microbial communities impact biogeochemical cycling. I'll be focusing can I go back? Sorry. My project mostly is focusing on this carbon cycling. So through respiration, microbes can release CO2 into the atmosphere, which is a greenhouse gas. You have specialized microbes, which are methanogens, that produce methane and release that in that atmosphere. And that methane can be oxidized either abiotically or by methanotrophs and release CO2 into the atmosphere. And Keiko will be going over, right after my presentation, some of the things we're looking at with the nitrogen cycle. Okay, I'm good for the next. So what I do is I collect soil samples to measure activity and diversity of the microbial community. If you look at this graph A, this is a measurement of activity of the microbial activity. So if they're actively reproducing, and generating energy. And what we found is that at Yampa, the microbes were more active than in Hester. So they all fall to the left of this 5%. EAF is just a measure of activity. They all fall to the right of that line, and Hester, the most active microbes, fall to the left. And if you look at B, we had a total of about 19,000 unique ASVs, which you can think of as species, and you'll see there's a higher, species richness at Hester. There's almost 6,000 unique ASVs there. And part of that is because when you introduced all the new soil on top to raise up the elevation, you're introducing a new microbial community that's now meshing with the existing microbial community, which increases that diversity. And we're also looking at biomass right now, where you're seeing that Yampa has higher biomass, as observed using microscopy. And the way we figure out biomass is if you look at this picture in the bottom right corner, basically we use microscope slides and we take pictures and we can select individual cells, which are in yellow, which are the little microbes, and we can select the chloroplasts. Basically, we are able to look at the specific diatoms and other things that are autotrophs in the site, and we can calculate the biomass at each site. I'm currently going through all these samples to calculate biomass, but right now it seems that Yampa overall has higher biomass, which is expected. So these results suggest that post-restoration Hester is less active and differs considerably from Yampa, and that can rapidly change as the restored wetland population becomes more established. And we want to understand the underlying microbial processes across stages of restoration to inform restoration practices for the establishment of a community that increases carbon sequestration and reduces the release of, carbon dioxide and methane back into the atmosphere. And that's a photo of our team on the left.
Thanks, Leah, and go ahead, Keiko.
Hi, I'm Keiko. I am an undergraduate in Adina's lab, and I'm mentored by Leah, who helps me in this project. I look specifically at nitrogen cycling microbes in wetland soils, and I want to answer the question, how do different microbial communities contribute to soil nitrogen cycling and nitrous oxide emissions, which is a greenhouse gas, and compare them across stages of restoration. So by doing so, I start with DNA extractions, which can then lead me to identifying, quantifying, denitrifying, and nitrifying genes. And this is important because nitrification occurs when ammonia is converted to nitrite and nitrite into nitrate, which is the available form of nitrogen in plants, and denitrification occurs when nitrate is converted to gaseous nitrogen and emitted into the atmosphere, and it can also emit N2O, the greenhouse gas. Thank you.
Cool. Thank you. And now we're shifting gears, and Barbara, I will let you share your screen for your presentation about the carbon markets and carbon sequestration in how can we use that. And I will stop sharing.
Okay, wonderful. So we're shifting gears a bit from the science to the policy and finance. So I direct the Berkeley Carbon Trading Project at the Public Policy School at UC Berkeley, and I've been so lucky to be able to work with Adina and many of the people that you're hearing speak today in a study of how do current carbon offset programs, how effective they are at supporting wetland restoration, and how can they be made more effective at supporting wetland restoration. So I'm going to give a little bit of background on carbon offsets and how the market works, and then our research questions, methods, and findings so far. So the carbon market. Basically, the way carbon markets work is it allows an emitter who has an obligation to reduce their emissions, either a regulated requirement, such as under California's cap and trade program, or voluntary companies and cities and universities that take on voluntary commitments to reduce their emissions, such as a carbon neutrality goal. And it allows those entities to, in part, meet their climate targets by paying someone else to reduce emissions. So on the voluntary market, we're looking at the voluntary part because that's where wetland sits today. So the voluntary market is created by carbon registries. These are nonprofit organizations that have stepped in to develop programs to issue credits from climate mitigation projects that companies and others can use to meet their targets. Registries develop protocols which go sector by sector and define what projects are allowed to participate and how do you estimate the greenhouse gas benefits from each project. Projects are allowed to participate if they meet the requirements and they monitor and calculate their impacts according to the protocols. Those projects then go through a third-party auditing system, and the registries issue credits according to those calculations, the monitoring over time and the calculations, and then companies and others can retire those credits when they use them so that they can't be used again by someone else. Taking a look at the market, we see, just to give you a sense of what project types are out there, over a third of the credits are from forestry, mainly this avoided deforestation, improved forest management, reforestation. Second most important is renewable energy and hydropower. You have improved cook stoves projects in the global south, and other household efficiency projects. You have chemical processes like refrigerant destruction, landfill gas capture, and other smaller project types. So and one thing to see here is that wetland restoration is one of these really small ones. So the market is not yet supporting wetland restoration at scale. So looking at other project types, we see to date, you could say 4 core quality challenges that have led to widespread overcrediting across that set of project types on the previous slide. And let me take as an example the avoided deforestation, which we've studied and is the project type with the most credits on the market. We see these 4 issues. Additionality, would the project have happened anyway? Are we just generating credits from what would have happened otherwise for other reasons? If the projects are additional, they wouldn't have happened. Well, what would have happened otherwise? And we see that avoided deforestation projects often have exaggerated baselines where they exaggerate the deforestation risk and the likely amount of forest loss that would have occurred without the project. We see issues of carbon quantification, such as exaggerating the amount of carbon per acre in the forest above ground and below ground. And we have unintended negative outcomes. For example, when you have a conservation area where forest communities are limited from their use of the forest, sometimes without their consent. So turning to wetlands, our question was, given the quality challenges, we asked 2 overarching questions. One is, how can wetland restoration offset programs effectively and more effectively, because we don't see many projects to date, effectively support wetland restoration with a focus in California? And 2, do the current protocols accurately estimate project climate benefits? And where they don't, how can they be updated to be brought in line with the science? And the methods that we use are basically we compare the protocols against the latest science, as well as the insights from practitioner interviews, looking across the major quality issues, additionality baselines, carbon quantification, and the broader effects of these projects. Key findings so far, and this is my last slide, is, question number 1, are offsetting programs effective? We find, or can they be effective if prices go up? And we find that for wetlands in California that have already been restored, what we see is that current funding sources, grant funding, often does not allocate funds for ongoing maintenance. So offsets could be a really good match for these funding needs because the offset income comes over time that can be used over time for the maintenance as is needed. But for new restoration, it's challenging because funding is needed upfront for reductions that accrue over time. And the costs are high per credit, per ton mitigated. And for restoration projects, we see a real potential for major buyers of credits or to contribute funds upfront through a contributions approach rather than buying offsets over time. Part of our recommendation to shift to contributions comes from the second part, that is, do offset programs accurately estimate project benefits? And we identified several key uncertainties that need more research for refined estimates. And one of those is what Aliya presented, that is on lateral flows of carbon outside of the wetland, where we really have there's still a lot of uncertainty about whether that's released or sequestered. And then we recommend several specific modifications to align the current protocol with the latest science and bring it up to date. And there you go.
Thank you, Barbara. And let me share my last slide here. Just a second. Share. Okay, so my last slide, I'm just trying to summarize what we know of what we heard so far from all of the talks. We know that wetlands can remove CO2 from the atmosphere and store the carbon in the soils for decades to millennia, which is a good thing. So we should conserve and restore wetlands. So, as I said, wetlands restoration can be effective nature-based climate solutions. We know that the sequestration in soils depends on many environmental conditions. And when I say sequestration, I mean the net sequestration, accounting for the emissions and the loss would depend on many, many different environmental conditions. And therefore, we need to actually understand what these conditions are in order to both do the mass balance and understand how much is sequestered, but also to inform restoration projects that want to maximize these CO2 emissions. And just a short thing about the potential or the possibility of including this in the carbon markets that will help both fund these kind of projects, but also that requires the monitoring, so it can help both quantifying the removal and getting money for important restoration projects. And with that, we're done here. And if there's questions, we'll all be happy to answer them. And I will stop sharing. And Monique, do you want to go ahead and get us started? Monique, do you want me to read the questions from the chat, or do you want?
Yeah, go ahead. I didn't realize I was muted. Yeah, sure. Go ahead and read the questions for your group.
Okay, so the first one is: does biochar raise the levels of methane metals in the soil? Well, this is something we're looking into. We don't know. There's data from previous studies where this is used in agriculture soil that sometimes show release of methane when it's under reducing of metals when the conditions are reducing, for example, in aquifer recharge, and sometimes it actually absorbs the metals. So we did not analyze yet the metals, but we will. That's part of the plan to do. Different types of biochar. Yes, we use right now two types of experiments. One is with walnut shells and walnut biochar, and the other one, which is somewhat more relevant to Elkhorn Slough, is they're using eucalyptus and removing eucalyptus. So this has a double benefit. It's a place to put all of this eucalyptus biochar and also mulch into the wetlands, and that's a new experiment that we just started. I don't think that biochar is likely to affect salinity. This is a very saline, open to the ocean wetlands, and I don't think the amount of biochar or the capacity of absorption of sodium by the biochar can have any impact on salinity. And okay, I think this one is about the methane emissions, and I assume that's from Pelican Marsh. The methane emissions are very, very low, even in Pelican Marsh, compared to freshwater systems, but it's still higher than in Yampa and in Hester. And part of the reason for that is that the water level there, it's always inundated and it's always anoxic, so there's no way for oxygen to get replenished there. there's a lot of organic matter, so we see high methane emissions from that site. But still, because of the salinity, it's less, the concentrations and the fluxes are lower than in freshwater systems. Okay, that's a question for JJ about why is the methane trend downwards with time, and that's for Hester. Do you want to answer that, JJ? Come on here.
Well, I don't know if I have a great answer. We're in the works. I haven't started my PhD yet, but it's possible that it is an outcome of our gap filling, where we had the tower fall over at the beginning of 2023. So there might be some issues with data, but if it's accurate, it could potentially be the downward trend of methane since restoration. But yes, yeah, specifically because methane is dominated by microbial processes, it would relate to the, how the microbes have become more
established.
Yeah.
So it really depends, as both JJ and Leah mentioned, on the microbial community. So if you have organisms that can oxidize this methane and they become established, there's less emissions of methane over time. So combining all these pieces of work will help us understand why there is a reduction in the methane. Marco, you go ahead. That's your next one.
I've already answered.
And okay, Marco already answered. People can read that. In Leah, did you compare? She's also already answered. Monique, will these answers will be available for people if they want to go do that later?
Yes, we'll be saving the chat for sure.
Perfect. So it looks like Leah answered the questions here. Okay. Aliya also answered. Do you want to answer again, or?
Sure. I'm happy to talk about it a little more. Emma brought up a good point that one of the unaccounted for types of carbon that is likely being exported and imported is macrodetritus and stuff like leaf litter and just like stuff that can be washed off from the surface into the marsh, or maybe created in the marsh if it's plants that are growing in a channel that die and then export to the ocean. From what I found so far, this is really not studied very well in salt marshes, or it may have been studied, but in the context of stuff like feeding coastal ocean food webs, and it hasn't been rectified well with this carbon budget. But people are definitely starting to think about it, so that's something that I'm starting to think about, and it's something that I see people do a lot of research on, especially in mangrove ecosystems where you have more stuff like leaf litter and detritus that is more obviously connected to the ecosystem that you can track out to the ocean, but it can be more challenging in salt marsh ecosystems. But yeah, it's a good point. It's definitely unaccounted for and something that's not generally mentioned in review papers about lateral carbon exports.
Cool. And then there's a question for you, Barbara. Do you want to answer it to everyone?
Sure. Yeah. So Kyle asked if for financing, have we thought about stacking carbon benefits with the range of other benefits from wetland restoration? And the answer there, it was too much for me to write, is that there already is a lot of funding going into wetland restoration for its range of benefits. So that gives a challenge to us to think about the issue of additionality, that how do you think about, so if there is stacked benefits and stacked financing, and then you add offsets into that, is the entire project able to generate credits and considered additional, or do you want to, how do we think about how the offset income has an effect on the outcomes, and to generate credits really in proportion to the money provided? And the answer that we've come to, or that we're considering, is payment stacking, where if other sources of funding provide, for example, 60% and offsets provides 40%, you can take responsibility for 40%. You as the offset purchaser can take responsibility for 40% of the carbon credits. And that's one possible answer to that question, and there are other ones as well.
Well, I don't see any more questions, but if anybody wants to follow up with questions with any of the students that presented here or Barbara, you can find our emails and information on the lab webpage and on the slides, so you can contact us later.
Thank you, Adina. Thank you so much. And we'll also be sending out a list of all the speakers and their contact information, so all of that will be in there as well. Just a fascinating session. So much work going into the blue carbon and such a fascinating area. Carbon sequestration is definitely one of the ecosystem services that we value in the Slough and particularly at Hester. And I think one of the things that we think about is how rare this habitat is. We've lost 80% of it within California, and we worry about losing it with sea level rise. So I think we're okay with the slow rate of carbon sequestration here. We're really taking that long-term view that we want to make sure that we have high marshes that are as resilient as possible to sea level rise. And so we know we're just going to have some patience in all of the plants coming in and all of those below ground processes. But definitely want to thank you for all of those great talks. And really thank everybody. This is the end of our session of our symposium today. And thank you all for coming and for all your input along the way. It's really meant so much to us to be able to share what we're trying to accomplish and what we've learned about this really special place. And our recording of all of this will be available soon. So thanks again for joining us.
Thank you both for coordinating that, Monique and Kirsten.
Thank you, everyone.